Okay, this is the brief on cognitive complexity, why really simple roles can actually hurt your brain, and what the number 147 has to do with stability. Ever feel forced into a box? Made to think too simply? Well, this material confirms a key idea. When you're pushed into just one narrow role, cognitive flattening, we call it really disrupts your biology, even your immune health. All right, let's break down the three big takeaways here. First, the sources confirm your stability needs constant biological chatter. Think mirror neurons, key for empathy, and your immune T cells. They have to communicate across systems. This whole integrated function, way beyond just one brain area, is critical for mood and resilience. Second, that cognitive flattening feeling, it's a real biological disruption. Your system is built for parallel integration, doing many things at once. Being forced into one simple role disrupts that. This lost connection even changes your immune tone, messing with T cell signaling, which can leave you feeling agitated or depleted. Finally, this need for complexity leads to the number 147 as a symbol for multidimensional stability. Picture it. The 1 is the foundation of independence, the 4 is the working platform of pragmatism, and the 7 is the capstone of wisdom. And the force linking them all, that's creativity. So, the biology really backs this up. It's complexity, not forced simplicity, that you genuinely need for mental and, well, whole system stability.
Welcome to the debate. Our discussion today, well, it really centers on something quite profound that's emerging from recent work on psychological distress. And that's the deep biological basis of this profound discomfort people experience. We're looking at how external pressures, social, relational, can kick off a physical response that then gets sort of stuck, sustained internally.
Hmm.
And the focus is this phenomenon called cognitive flattening. The sources we're looking at confirm it's this fundamentally disruptive state where the body is almost forced into this narrow, kind of linear way of processing what should be complex multidimensional input.
Right. And the crux of our debate today is figuring out the primary factor that keeps this flattening going. We both agree the system's dysregulated, but what's sustaining it? Is it the constant moment-to-moment feedback loop of social misrecognition, like ongoing role compression, essentially, the persistent external environment?
Or is it something deeper? Is it the entrenched chronic biological changes, specifically thinking about these microbiome legacy effects that could be initiated by, say, prior medication use, that has fundamentally reset the internal system, keeping the dysregulation going almost regardless of what's happening outside right now?
Exactly.
So I'll be making the case that this sustained feedback loop of, let's call it, social misrecognition or flattening is really the primary engine here. It's what keeps the entire biological system, immune function, the HPA axis, even neural networks in this state of chronic high alert.
And I'll be arguing the other side, that the persistent chronic biological alterations, especially those linked to the gut-brain axis, the GBA, and this idea of immune memory, well, that these create an underlying entrenched vulnerability. And this vulnerability basically dictates the system's susceptibility. It acts as a kind of permanent substrate that sustains the cognitive issues and mood problems, even if those external stressors lessen for a bit.
Okay, so my core position really starts from the idea that, well, the organism, the body, it's built for complexity. You have to think of it less like a single engine and more like a symphony, right? You've got neural, immune, hormonal, chemical systems all playing together, adapting fluidly to context. That's what I'd call parallel integration. Now, when the external world imposes what the source calls a linear reduction, demanding a person fit into one single rigid role, maybe ignoring their depth, that symphony gets muted. It's forced to play just one simplified, maybe frantic note. That forced compliance is cognitive flattening, and I'm saying it's inherently biologically disruptive, even traumatic.
Okay, and how does that happen biologically, in your view?
Well, the source material lays it out pretty clearly. It triggers an immediate neuroendocrine alarm. The limbic system basically flags this simplified, rigid social input not as safety, but as a threat signal. So the HPA axis kicks in, right? Floods the system with cortisol. And this chronic stress, it doesn't just feel bad, it physically changes immune tone. Microglia can become reactive, inflammatory, T-cell signaling shifts. This whole physiological struggle forces a downregulation of those associative neural networks, which leads to that subjective feeling of emotional pain, exhaustion, the loss of color and thought people describe. So for me, the chronic stress that comes from neglect or misframing, that's the critical sustaining input. It means that recognition isn't just, being nice. It's actually a biological intervention. It lowers the threat output, helps stabilize those hormonal and immune rhythms.
Okay, I see the argument for how stress, especially social stress, initiates distress. It's compelling, but I think it perhaps undervalues the sheer depth and, frankly, the longevity of the biological changes that can occur internally. So I come at it from a slightly different angle. For me, the core issue, the thing that primarily sustains the problem, is that chronic biological alteration, specifically to the gut-brain axis, often driven by pharmacological history.
Right, the medication angle.
Exactly. We know, and the sources confirm this, that many common medical drugs, things like SSRIs, say fluoxetine, antipsychotics like olanzapine, even drugs people might not think of like proton pump inhibitors, omeprazole for acid reflux, they can significantly disrupt the gut's delicate ecosystem. And crucially, the point the sources really hammer home is that these disruptive effects can persist long after someone stops taking the medication. This creates what the literature calls a microbiome legacy effect.
A legacy effect. Okay.
Yeah, and it's not just that recovery is slow. It's potentially a completely recalibrated operating system for the gut and therefore the brain. The persistence seems to be driven by, three main factors outlined in the material. First, epigenetic changes. Drugs can actually trigger long-term gene expression modifications in gut cells, immune cells. Things like mRNA 193 regulation are mentioned. Second, immune memory. T cells and microglia, they essentially remember past inflammatory states, staying primed, sometimes for months or years, which maintains this low-grade neural inflammation. And third, there's neural conditioning of the gut's own nervous system, the enteric nervous system. So you put these together and you get this hypersensitive biological baseline. What often looks like an emotional relapse or this persistent cognitive flattening we're talking about might actually be the system just operating on this fundamentally shifted microbial and immune foundation, almost independent of the immediate social stuff.
Okay, let's maybe dig into that immune point, because I think that's where our positions really diverge, isn't it? You're right, of course. Acute social stress modulates immune tone through cortisol. But your argument is that if the threshold for that immune response is already lowered, if the system's been primed by, say, past medication, doesn't that permanent readiness become the main issue, the thing that dictates how the system handles any stress, social or otherwise?
Precisely. I'm arguing that the priming itself, that legacy effect, is the primary problem. It's like, imagine you installed a super sensitive hair trigger alarm system because of the medication. Now you can get rid of the installer. The drug itself is gone, but that hair trigger mechanism remains. So now every time the door creaks, metaphorically speaking, the whole system just explodes in inflammation. The current social input might just be that accidental door creak. The real engine sustaining the problem is the faulty alarm system that was installed maybe months or even years ago.
That's a useful metaphor, the hair trigger, though I'd frame its significance a bit differently. Yes, that biological memory, that priming, it certainly exists, I don't dispute that. But its activation still seems to be immediately and constantly modulated by the current environment. A prime cell still needs a signal to fully activate, right? So if the social environment changes, if genuine complex recognition actually happens, that recognition acts as a direct biological input. It should, according to the sources, drop the threat signal, lower amygdala output, reduce cortisol.
But does it? Sufficiently?
Well, that's the point. It provides an immediate instruction for those primed immune cells to stand down.
A hair trigger alarm is only dangerous if the trigger is constantly being pulled. So if chronic social misattunement is that perpetual pulling of the trigger, then that remains the factor sustaining the inflammatory cycle. It overrides the system's ability to stabilize, even with that underlying biological vulnerability.
But that assumes the only reason the trigger's being pulled is current social misattunement. What about the sheer physical discomfort associated with the state? You know, the fatigue, the cognitive fog you mentioned earlier. This leads us right into the second key point. What is this cognitive deficit really?
Okay, yeah, so I define that cognitive flattening as this disruption of the normal, sophisticated crosstalk between key brain networks, prefrontal cortex, limbic system, mirror neurons, places like that. It happens because of forced compliance, like we discussed. The brain is geared for, expects complex reciprocity, nuance, but the input it gets is rigid, maybe authoritarian, overly simplified. And this forces those associative networks to downregulate. The result is that subjective feeling of losing color or dimensionality of thought. That feeling of dullness, constriction, it's directly tied to that neural network misalignment driven by the social context. It's almost like an injury to the meaning-making capacity itself.
I'm sorry, but I struggle with the idea that the persistent physical fog, the fatigue, the irritability, that whole constellation of symptoms is solely a failure of neural architecture or meaning making. Let me explain why. That entire cluster of symptoms is frankly perfectly explained by a physical illness originating from the GBA legacy effect we talked about.
How so?
Well, the literature is clear. Systemic inflammation driven by dysbiosis-induced cytokines, things like IL-6, TNF-alpha, these cross the blood-brain barrier. They cause measurable cognitive impairment, mood shifts. This isn't symbolic, it's metabolic missignaling. Gut hormones like ghrelin and leptin, which are critical for appetite and energy regulation, get missignaled. That leads directly to fragmented sleep, maybe severe appetite shifts. So the cognitive effect, the fatigue, the brain fog, the inability to focus, it looks like a downstream symptom of this underlying physical dysregulation. The system is in a way physically sick from that microbial disruption, and the cognitive flatness is just the brain expressing that foundational sickness.
Okay, the systemic inflammation, the cytokine signaling, that's undeniable, absolutely. And those are critical consequences, I agree. But we still have to address the mechanism of perception. The sources also confirm that the brain interprets all this physical discomfort through what they call interoceptive prediction loops. Essentially, the brain's constantly trying to guess its internal state, its sense of inner balance. Now, when dysbiosis is causing chaotic signals from the gut, and the social environment is simultaneously confirming you are unsafe, your complexity isn't welcome here, the system reinforces the prediction that the internal chaos is the external threat made manifest. So the failure, I think, is one of integration. That feeling of flattened cognition isn't just the cytokine fog. It's the inability of the brain's architecture to integrate those chaotic physical signals into a coherent, safe narrative. Precisely because the social feedback loop keeps insisting that complexity and safety just cannot coexist for this person.
Okay, which brings us logically to our final and maybe most practical point, the path to regulation. If the driver is this entrenched microbial legacy, as I'm maintaining, then the path to recovery really has to start with biological repatterning. We need to supply the physical infrastructure needed for stability first.
Meaning?
Meaning things the sources suggest. Practical systemic interventions. Supplying things like prebiotics, polyphenols, from certain foods to encourage the recolonization of beneficial gut species. Performing mind-body coherence practices like specific breathing exercises to restore vagal tone. Maybe monitoring thyroid and adrenal rhythms. We have to directly correct that permanently shifted baseline flora and the systemic inflammation it causes. If the gut itself is constantly producing inflammatory signals, then just addressing the neural network crosstalk feels a bit like repainting a house while the foundations are rotting.
I definitely see why you prioritize that foundational physical layer. It makes sense. But I would argue that real, lasting healing needs more than just physiological support. It requires cognitive symbolic integration, perhaps even first, or at least concurrently. We have to retrain those interoceptive prediction loops you mentioned.
But how, if the physical signals are still chaotic?
Well, your suggestions, the prebiotics, the vagal stimulation, they're absolutely crucial support mechanisms, no question. But I think they're unlikely to achieve long-term balance if the environment keeps sending the message, your complexity is fundamentally unsafe. The amygdala's threat output will likely stay high, the cortisol response will continue, and that will prevent the stabilization of the very hormonal and immune rhythms needed for your physical repatterning to actually stick. The system needs to learn, deeply learn, that complex safety is possible again. And that learning happens through receiving valid, multidimensional recognition, which is fundamentally a cognitive and symbolic process that breaks that negative social feedback trap.
But you risk falling into the opposite trap, don't you? Trying to use cognition to solve what might be, at its root, a fundamental physical problem. When we look at the mechanisms described, those epigenetic changes, the long-term impact of miRNA 193 regulation on cellular memory, we're talking about really persistent, deeply entrenched biological shifts. This isn't just about managing immediate stress.
If the microbial composition has truly, fundamentally changed the baseline flora, and the gut's own nervous system is conditioned to this sort of hyperactivity, then the individual is likely to be prone to inflammation and internal chaos almost irrespective of how many positive social cues they receive externally. We surely have to address that biological infrastructure first, or at least give it equal weight. Otherwise, the cognitive integration you're seeking might be built on shifting sands, leading only to more vulnerability when the next inevitable stressor comes along.
So what we've really confirmed today, I think, is the absolute intricate inseparability of the body's symbolic, emotional, and microbiome ecologies. They are deeply intertwined.
Indeed. And we acknowledge the powerful evidence for that GBA disruption, the immune memory, the epigenetic effects potentially stemming from pharmacological history, all creating this deep substrate of vulnerability.
However, I still maintain that the active, perpetuating engine, the factor that really amplifies that vulnerability and prevents the system from finding a new balance, is that chronic, pervasive stress generated by social misattunement or flattening. That failure to gain recognition triggers a continuous high alert status, keeping the whole cycle locked firmly in place.
And I would reaffirm that the sheer depth of that biological legacy, the potential for permanent shifts in microbial composition, the entrenched immune memory, means that this biological state fundamentally dictates a system's susceptibility and its capacity for finding homeostasis again. This entrenched baseline suggests that biological repatterning is the essential, perhaps primary point of intervention, determining whether any cognitive or social integration can actually take hold and succeed long-term.
It seems understanding this multi-scale complexity is absolutely necessary to figure out how to restore equilibrium. The question maybe for the listener to ponder is where the most effective initial intervention lies. Is it in resolving that immediate, chronic social stressor through achieving symbolic integration?
Or is it in repairing the entrenched biological vulnerabilities, perhaps inherited from pharmacological history first? Where do you start untangling that knot?
Welcome to the deep dive. So today we're tackling something, well, it's pretty sophisticated, isn't it?
It really is. We're bridging some complex fields here.
Yeah, this hypothesis connecting, neurogastroenterology, psychoneuroimmunology, and even the social aspect. And the core ideas are challenging. First is that feeling of social neglect or being kind of forced into a narrow role, what you're calling cognitive flattening. Is that actually a form of biological trauma? And then the second piece, can stopping powerful medications leave this lasting disruptive blueprint on your body, on your core systems, long after the actual drug is gone?
And look, that intuition is remarkably sharp. This isn't just some vague theory anymore.
Yeah.
It's really where cutting-edge systems biology is starting to align with lived experience. Your sources laid out a clear framework, and the science is definitely catching up.
So you're saying the premise holds up, that this flattening is biologically disruptive, and the medication effects linger through the gut-brain axis?
Absolutely. It's completely valid, and I'd say increasingly well-supported scientifically.
That's That's powerful validation right from the start, because our listener, you, want to understand the scientific nuts and bolts behind this feeling, the how and the why the body seems to hold on to this altered, maybe more vulnerable state.
Exactly. And that's our mission today. We're going to trace that biological legacy.
Okay.
We'll start with the foundation, this constant conversation happening between your brain and your gut. Then we'll get into how common drugs can kind of hijack that system.
Right.
And then we'll detail the specific ways, molecular, cellular, that the body maintains that disruption. And finally, importantly, we'll connect it back to how that external social pressure, that flattening you described, can actually trigger this already primed system into a state of, well, chronic trauma.
Okay. Let's dive in. Section 1, the gut-brain axis. So let's kick off right at the heart of this. The gut-brain axis, the GBA. You know, people often think the brain's just barking orders down, right?
Yeah, the old top-down view.
But the sources describe it very differently, like a constant two-way street, a master circuit, always sending signals back and forth.
That's spot on. The word axis is really key here. It implies these two major hubs connected by, well, communication lines. We're talking about the link between the central nervous system, your brain, and spinal cord, and the enteric nervous system, the ENS, which is this incredibly complex network of neurons lining your gut.
The second brain, right, I've heard that term.
Exactly. It's earned that nickname because it's so extensive and can operate with a surprising degree of independence. It manages digestion, obviously, but it's also constantly signaling the state of your internal world, your gut ecology, straight up to your head brain.
And this isn't just one signal path. The sources mentioned multiple distinct communication highways keeping the gut and brain in sync.
Right. There are three main ones we should probably break down.
Let's do it.
Okay, first you've got the most direct route, the fastest one, the vagus nerve.
Ah, yes, the vagus nerve.
And think of it like a major fiber optic cable running directly from your brainstem down into your abdomen. It allows for really rapid neural signaling.
So, like, when you feel butterflies in your stomach, that's the vagus nerve talking.
That's a perfect example. Or when your gut microbiome senses something's off, maybe inflammation or imbalance, the vagus nerve makes sure the brain gets that feedback almost instantly. It's the physical pathway for interoception, that sense of what's happening inside your body.
Okay, direct line. What's the second highway?
The second one brings your entire defense force into play, the immune system. This is where the health of your gut microbes really translates directly into your neurochemistry.
Okay, hold on. How does that work exactly? Gut inflammation affecting the brain. Isn't there the blood-brain barrier keeping things separate?
That's a really excellent question.
Yeah.
And it gets right to the core of, well, psychoneuroimmunology. The blood-brain barrier is protective, definitely, but it's not completely impenetrable, especially not to chemical signals.
Oh.
See, your gut houses something like 70 to 80% of your body's entire immune system. It's huge.
Wow.
So when your gut is stressed, or you've got dysbiosis and imbalance in microbes, these gut immune cells release signaling molecules. They're called cytokines.
Cytokines, those are the inflammation messengers, aren't they?
Precisely. They're like little molecular messages, chemical communiques. And these pro-inflammatory cytokines, they travel through your bloodstream. Now, the bigger immune cells, like T cells, might not cross that barrier easily.
Right.
But these tiny cytokine messengers, they can cross or they can activate receptors on the barrier itself. And that prompts the brain's own local immune cells, the microglia, to react.
So it's like the gut is phoning ahead, telling the brain's immune system to get ready.
Sort of, yeah. It directly links inflammation in the gut to potential inflammation in the brain, neuroinflammation. And that fundamentally shapes your mood, your energy levels, your cognitive function.
Okay, so immune signals are relaying the gut's status. What's the third communication route?
The third major one is the endocrine system. This is all about hormones. Think of it as setting the long-term chemical background music for the whole gut-brain conversation.
Hormones like cortisol, stress hormones.
Exactly. When you experience external stress or even internal dysregulation, the HPA axis, that's the hypothalamic pituitary adrenal axis, kicks into gear.
The stress response system.
Right. It floods your system with stress hormones like cortisol and ACTH. And here's the thing. These hormones don't just impact your brain. They act directly on your gut lining, too. They modify how the immune cells in the gut talk to each other, and they can literally change the gene expression and the barrier function of the cells lining your gut.
So stress creates this signature that's felt equally in the gut and the brain.
Absolutely. It's a system-wide response.
And the big takeaway from this constant three-way dialogue, it seems to be most obvious when we look at neurotransmitter production. The sources mentioned staggering numbers.
It really is the most compelling statistic in this whole field, I think. Roughly 90% of your body's serotonin is actually produced in your gut, not your brain.
90%. That's incredible. Serotonin for mood, stability.
Well-being, sleep cycles,
Yeah.
yes. And on top of that, a very large portion of the precursors for dopamine, another key neurotransmitter, are also made in the gut.
So what does that mean, fundamentally?
It means there's this inherent vulnerability. Any significant shift, any imbalance, any disruption to your gut microbiome, it doesn't just cause maybe some digestive upset. It immediately and fundamentally influences your mood regulation, your cognitive clarity, and your basic ability to handle stress. The gut is acting like the nervous system's chief chemical engineer in a very real way.
That sets the stage perfectly for the next part. If the GBA is so central and sensitive, what happens when medications enter the picture? Okay, knowing how crucial and, frankly, delicate that gut-brain balance is, let's pivot to your experience with medication. It does seem a bit counterintuitive, doesn't it? That drugs designed maybe for the brain, or even something simple like an acid reflux pill, could act as these profound, lasting disruptors in that gut ecosystem.
It does seem counterintuitive, I agree, until you really appreciate that the gut is this teeming, open microbial world, and most medications are, well, bioactive molecules. They interact with things.
Right.
So the general principle that's really emerging from the research is that many common drugs, psychiatric ones, but also others, they interact directly with gut bacteria, or they change the pH balance in the gut, or they alter the mucosal lining. And the result isn't always temporary. It can lead to a persistent microbial signature.
A microbial signature, like a lasting fingerprint on the gut flora.
Exactly. A lasting change in the ecosystem's composition and function.
Okay, let's get specific then. The sources listed several types. Let's start with the ones often used in psychiatric care. SSRIs and SNRIs, like fluoxetine or venlafaxine.
Right. So these meds are designed to boost serotonin availability in the brain. We know that. But remember where most serotonin lives?
In the gut. 90%.
Precisely. So these drugs inevitably have potent effects there too, even if unintended. First off, many SSRIs actually show direct antimicrobial activity.
Wait, they kill bacteria?
Some of them, yes. Or they inhibit the growth of certain species while letting others take over. This alone can shift the balance, create dysbiosis. Secondly, some research indicates they can increase intestinal permeability.
That's the leaky gut idea again, isn't it?
That's the mechanism, yes. The tight junctions, the seals between the cells lining your gut, can become compromised.
And when that happens?
When the barrier gets leaky, stuff gets through that shouldn't, maybe partially digested food bits, bacterial fragments called endotoxins. These leak into the bloodstream and can fuel low-grade systemic inflammation, which, as we discussed, can reach the brain.
Okay. And SSRIs might also mess with bile acid metabolism.
Yes, that's another pathway. Bile acids are crucial for regulating gut flora, and SSRIs can interfere with how they're processed, adding another layer of potential disruption.
Okay, moving on to antipsychotics. Things like olanzapine or risperidone. These often come with really significant metabolic side effects like weight gain. Where does the gut fit in there?
Antipsychotics are known to be quite strong microbial disruptors. Research shows they often cause a really significant shift in the types of bacteria present in the gut.
How so?
They tend to favor bacteria that are, let's say, very efficient at extracting calories from food, even from diets that aren't necessarily high calorie.
Ah, so the gut microbes themselves contribute to the weight gain.
That's strongly implicated now, yes. This microbial shift seems to be a major driver behind those recognized side effects. The rapid weight gain, the insulin resistance, and also potentially increasing low-grade neural inflammation. The change of the gut environment really seems to dictate the metabolic outcome for many people.
We definitely need to talk about antibiotics, especially broad-spectrum ones. Even just one course can have huge effects, right?
Oh, absolutely. Antibiotics are essentially like setting off a bomb in the gut ecosystem.
Yeah.
A biological scorched earth approach. They cause this massive, immediate wipeout of microbial diversity.
And the key point for the listener, for this idea of a legacy effect.
The key is that recovery, the recolonization, it's not a quick bounce back. It's slow. It's nonlinear. It can take months, sometimes even years, to get back to something resembling a stable state.
And maybe never fully back to the original state.
For some people, yes, especially those who might already have a sensitive system, maybe due to genetics or neurodivergence. The new flora that eventually establishes might be a permanently shifted baseline, a new ecosystem that's maybe less diverse, less resilient than the original one.
Wow. Okay, the list and the sources continued. What about systemic hormones, like thyroid medication or corticosteroids like prednisone? People take those for all sorts of things.
Right, these drugs modulate the body's master control systems. Corticosteroids, for example, are designed to suppress the immune system. But in the gut, they directly affect the gut's own immune cells and the cells lining the gut. They can change mucosal gene expression. So when you shift the body's overall metabolic or immune set point with these powerful drugs, the gut environment has to adapt its own inflammatory and regulatory settings. This contributes to a lasting change in its functional state.
And even common over-the-counter stuff like proton pump inhibitors, PPIs for acid reflux, like omeprazole.
Yeah, don't underestimate those.
Yeah.
PPIs fundamentally change the physical environment of the upper digestive tract. Stomach acid isn't just for digestion, it's a crucial barrier. It kills off a lot of bacteria we swallow.
So less acid means?
Less acid means more bacteria survive the trip down. This can lead to something called SIBO, small intestinal bacterial overgrowth. Basically, too many bacteria setting up shop higher up in the gut than they should be.
And that's a problem because...
It's highly problematic. These misplaced bacteria compete with us for nutrients. And critically, they can alter the availability of those precursor molecules we need to make vital neurotransmitters like serotonin and dopamine, again, impacting the brain through the gut.
So across the board, these drugs can create a dysregulated gut state. But if the system adapts to that state while the drug is active, why doesn't stopping the drug just hit a reset button? Why the persistence? Why this legacy effect?
Yeah, that's the million-dollar question. It seems to come down to the system establishing a new kind of sticky baseline. The microbiome, once it's shifted, it develops its own inertia. It kind of fights to maintain its new configuration.
And for people with already sensitive systems?
For individuals whose systems are perhaps already hypersensitive or highly reactive, and this often correlates with neurodivergence, that new, maybe dysregulated microbial signature can almost become the perceived normal for the body. The immune cells, the enteric nervous system, they get accustomed to this altered internal landscape. They start treating the dysregulated state as the default setting, and that makes recovery, getting back to a true baseline, really difficult and slow. It's not just about the drug leaving, it's about the ecosystem having changed.
Okay, so the ecosystem itself resists going back. That leads us to the deeper mechanisms. How does the body remember this altered state? Right, this is where we really get into the weeds, the technical side of your question, the idea of molecular memory. How does the body maintain that altered state, that microbial signature, even months or years after the drug itself is totally gone from your bloodstream? The sources pointed to 3 key mechanisms creating this biological legacy.
Absolutely. Think of it like the body's software has been subtly but persistently reprogrammed by the drug or the changes it caused. The first mechanism is really quite sophisticated. Epigenetic changes.
Epigenetics. Okay, that always sounds complex. Can you break down how a drug could leave a lasting molecular mark like that?
Let's try an analogy. If your DNA is like the hardware, the basic instruction manual for your body,
Okay.
then epigenetics are like sticky notes or highlighter marks on that manual. They don't change the fundamental instructions, but they tell your cells which instructions to read, how often, and how loudly to read them.
So they control gene expression.
Exactly. Now, drugs themselves, or the inflammation resulting from the gut dysbiosis they cause, can act as environmental signals that trigger these epigenetic changes. We see things like chemical tags called methylation getting added to DNA, or modifications to histones. Those are the proteins that package up the DNA. These changes can happen in the cells lining the gut or in the immune cells there.
So the drug flips a switch, changing how certain genes operate long-term.
That's a good way to put it. It rewrites the operating instructions for key processes, maybe things related to inflammation or barrier function. It leaves this molecular signature that dictates gene expression patterns long after the initial trigger of the drug is gone. For instance, the drug might have effectively told a cell, be on high alert from inflammation, and that epigenetic switch just stays flipped on.
And the sources specifically mentioned microRNAs, something called miRNA 193. How do these fit in?
MicroRNAs. These are fascinating. They're like tiny molecular dimmer switches for gene expression. They don't code for proteins themselves, but they bind to messenger RNA, the blueprint for making a protein, and control how much of that protein actually gets made.
So they fine-tune things.
Precisely. If a drug or the resulting dysbiosis messes with the regulation of specific miRNAs like miRNA 193, it can subtly shift entire regulatory pathways. It might keep inflammatory proteins slightly elevated or receptors slightly more sensitive, again, long after the drug is cleared. It's a very persistent, low-level way the system maintains that altered baseline.
Okay, epigenetic marks. What's the second mechanism for this legacy effect?
The second one is absolutely crucial for understanding why symptoms might persist. Immune memory.
Immune memory.
Yeah.
The immune system remembers being under attack.
In a way, yes. This is a major area of research right now. Your immune cells, particularly certain types of T cells in your body, and also the brain's own specialized immune cells, the microglia, they aren't passive responders. They actually retain a kind of memory of the inflammatory patterns they encountered while the medication was active or during that initial post-medication phase.
So it's like cellular PTSD. The immune system stays hypersensitive.
That's a really good analogy, actually. You can think of these cells as being primed, like hypersensitive smoke detectors. If the medication or the gut imbalance it caused triggers significant inflammation, these cells can remain in this high alert prime state for months, maybe even years. They're ready to overreact to even the slightest subsequent signal of stress or minor disruption.
And that constant readiness keeps things simmering.
Exactly. It maintains a state of low-grade simmering inflammation, including neuroinflammation, throughout the system.
And this isn't just theoretical, right? The sources linked this to actual brain changes in people with mood issues.
Yes, the connection is becoming much more tangible. Recent breakthroughs have actually identified persistent changes in gene activity inside specific brain cells in people dealing with chronic mood dysregulation. They found changes in a particular class of excitatory neurons involved in stress and mood.
Okay.
And, crucially, also in certain subtypes of microglia. This really helps confirm that this cellular memory linked to past stress or inflammation is measurable and contributes to the system's ongoing vulnerability after the drug is stopped.
Wow. So the immune system learns a lesson and refuses to stand down easily. That takes us to the third mechanism, which sounded more like learned behavior in the nervous system itself, neural conditioning.
That's right. This ties into the concept of interoception, the brain's ongoing sense of the body's internal state, what's happening inside.
Yeah.
When a medication is actively altering signaling, let's say it's artificially changing serotonin levels for months or years, the gut's own nervous system, the ENS, adapts to that new chemical reality. It learns to function within that altered environment.
So it gets used to the drug being there.
It does. Then, when the drug is stopped, the ENS suddenly finds itself with a communication system that's been functionally warped or recalibrated based on the drug's presence.
So the brain's internal map, its expectation of what balance feels like, is now based on a faulty setting. It's miscalibrated.
Exactly. The brain's interoceptive expectations can stay dysregulated. The whole nervous system essentially learned to operate on a foundation of distorted chemical input, and now it might treat that distorted signal, or the lack of it, as the baseline for normal function.
Which would feel wrong.
It would likely feel chronically wrong or unstable. Trying to operate from this miscalibrated baseline leads to that persistent unease, maybe anxiety, that fundamental sense that the body's internal environment is just off. Even though the drug is gone, the entire system has to effectively relearn what normal actually feels like without the drug's influence.
Epigenetics, immune memory, neural conditioning. 3 powerful ways the body holds on to the past. Now, how does this connect to that feeling of flattening? Okay, this is the critical link back to your core experience, the listener's focus on cognitive flattening. You describe your natural state as requiring this multidimensional focus, meeting parallel integration across all these systems, neural, immune, molecular, just to feel stable. And when that complex focus gets crushed by external pressure, you feel it as actual biological trauma. How does the science we've just discussed support that specific experience?
It supports it profoundly, I think. When you talk about a multidimensional focus biologically, that suggests a neural architecture designed for parallel integration. Your brain is likely wired to process multiple streams of information, simultaneously emotional, intellectual, sensory data, weaving them together to create a rich, stable, nuanced understanding of reality.
And that complex, integrated state feels inherently safe, right? It's the natural operating mode.
For that kind of nervous system, yes. But then, when social dynamics, or maybe the healthcare system, forces that intricate attention into a single, narrow, rigid channel, like being defined only by a diagnosis or treated solely as the patient.
Right, reducing you down.
That enforced simplification actively shuts down the parallel processing. The external pressure mandates linear reduction. Your neural networks involving the prefrontal cortex for higher thought, the limbic system for emotion, and crucially the mirror neuron system that handles social understanding and empathy, they're basically forced to stop their complex crosstalk.
And when the environment dictates that simplification.
The brain literally starts losing its dimensionality of thought. It can feel like the world loses color, depth. That experience is cognitive flattening, happening at a neural level.
So this social misrecognition, this external refusal to see or mirror your complexity, it's not just hurtful or invalidating psychologically. It registers as an acute threat that triggers a whole cascade, a neuroendocrine alarm loop.
That's precisely the biological reality of that kind of social invalidation, especially for a system primed for complexity. When the environment insists on treating you in a one-dimensional way, your nervous system registers a severe predictive mismatch.
A predictive mismatch, what does that mean?
Your brain is constantly making predictions about the world, including social interactions. It expects complex, dynamic reciprocity, especially if that's your natural mode, but instead it receives something rigid, simplified, maybe authoritarian. This mismatch is flagged by the limbic system, specifically the amygdala, the threat detector, and the anterior cingulate cortex, the ACC, which monitors conflict and errors. They flag this invalidation not just as unpleasant, but as an existential threat to your way of being.
And when the amygdala flags a threat, we know what happens next. HPA axis activation.
Instantly. The body floods with stress hormones, cortisol, ACTH. Now here's the crucial connection back to the legacy effects we just detailed. This acute stress spike from the social flattening, from being misunderstood or reduced, it doesn't hit a clean slate.
No, it hits the system that already has the GBA legacy from the medication, right? The primed immune cells, the epigenetically altered pathways, the conditioned nervous system.
Exactly. It hits primed microglia, T cells ready to overreact, and ENS already miscalibrated. So the social misattunement doesn't just cause a normal stress response. It causes a massive disproportionate chemical and inflammatory overload because the system is already sensitized.
So the stress from being flattened triggers the leftover biological signatures from the medication, reinforcing the instability. It sounds like a vicious cycle, a chronic feedback trap.
That's the absolute essence of the biological trauma you're describing. The social interaction provides the trigger, the flattening, the invalidation, which causes the hormonal spike, cortisol. This spike then activates the primed immune system. T cells misfire, microglia react because of the GBA legacy.
And the result of that internal chaos, the anxiety, the fatigue, the brain fog, how does that get interpreted?
Often, unfortunately, it gets misinterpreted by external observers, maybe even clinicians, as simply a relapse of the original condition, or inherent instability or treatment resistance, which then tragically confirms the initial misattunement's false, flattened assumptions about you.
While internally your higher brain is getting this terrifying message, your complexity is dangerous, it leads to pain.
Exactly. Your way of being is unsafe in this world.
That sounds absolutely exhausting. The sheer effort involved in fighting that internal and external pressure, that architectural narrowing, must be immense. What are the cognitive consequences?
It leads directly to the kinds of symptoms you mentioned. Losing the color or dimensionality of thought, feeling like your thinking is dulled or flat, and often a profound, almost dissociative fatigue. You are literally fighting your own neuroendocrine architecture.
How so?
To survive the simplified, threatening social exchange, your system is forced to actively downregulate the very associative brain networks responsible for complex thought, nuance, and flexible emotional processing. This effort to reduce your own complexity to match the environment's demands is what manifests as cognitive dulling and that deep fatigue. Your brain is actively suppressing its preferred complex operating mode just to cope with the perceived external threat. It's incredibly costly, biologically.
Which brings us to the daily reality of living with this, the functional consequences. Okay, having laid out that whole complex interplay, the GBA legacy, the social flattening trigger, the feedback loop, the clarity this brings, allows us to really reframe those persistent symptoms, things that might have felt inexplicable or purely psychological. Maybe they aren't. Maybe they're the direct, measurable fallout of this ongoing dysregulation. We need to start seeing these as potential microbiome legacy effects interacting with current stress.
That reframing is absolutely critical. It's empowering, and it points towards different solutions. If you recognize these issues, at least in part, as biological system imbalances rooted in that gut-brain axis history, you move away from just labeling them as a personal or psychiatric failing.
Right. So let's tick through some common ones mentioned in the sources, looking at them through this GBA legacy lens. First up, the almost universal experience for many people: persistent anxiety and fatigue.
Yeah, these are so common. The persistence here can often be traced back to that disrupted vagal tone we talked about. Remember the vagus nerve, that superhighway?
Direct line.
If the gut's enteric nervous system is still sending wonky, maybe subtly inflammatory or dysregulated signals up that nerve highway, even years later, the whole system could remain stuck in this chronic state of low-grade sympathetic nervous system activation, like a constant low hum of fight or flight.
Which feels like anxiety.
Exactly. It translates directly into persistent anxiety, maybe agitation, restlessness, and that chronic activation of the stress response system, the HPA axis, eventually just leads to burnout, that profound, often unexplained fatigue. Your body is simply exhausted from being constantly on edge.
Okay, that makes sense. Then there's sleep disruption, fragmented sleep, difficulty staying asleep. That seems almost inevitable if the gut is making 90% of the serotonin precursors.
It follows directly, doesn't it? Serotonin, mostly made in the gut, is the essential precursor molecule for making melatonin.
The sleep hormone.
The main sleep hormone, yeah. Melatonin regulates our circadian rhythms, our sleep-wake cycles. So if that GBA legacy has compromised the gut's ability to synthesize serotonin efficiently, or maybe absorb the tryptophan needed to make it,
Right.
then the body simply doesn't have the raw materials it needs to produce adequate melatonin to orchestrate deep, restorative, coherent sleep. So you see fragmented cycles, trouble falling asleep, trouble staying asleep. It's often a direct manifestation of this gut-derived chemical imbalance.
Many people also report really significant metabolic shifts after taking or stopping certain meds, especially as you mentioned antipsychotics, but maybe others too.
Yeah.
Changes in appetite, weight.
Yes, this is a classic example of the GBA legacy playing out. We know dysbiosis, that altered gut microbial community, directly changes the signaling of key gut hormones that are supposed to regulate metabolism and appetite.
Which hormones are we talking about?
Primarily things like ghrelin, which is often called the hunger hormone, and leptin, the satiety hormone.
Yeah.
When the microbial environment is significantly shifted, it can lead to these hormones being missignaled. Maybe you feel hungry when you don't need calories, or you don't get the signal that you're full. This contributes directly to appetite dysregulation, potentially leading to weight changes, shifts in metabolism, and sometimes downstream issues like insulin resistance. The gut's altered chemistry is literally dictating systemic metabolism.
And lastly, that pervasive, frustrating feeling of cognitive fog or irritability, like your brain just isn't firing on all cylinders and your fuse is short.
Yeah, this symptom is very strongly linked to that low-grade neuroinflammation we keep coming back to.
From the cytokines.
Exactly. When the gut remains dysbiotic or the gut barrier is leaky even years after the initial drug exposure, the immune cells lining the gut might continue to release slightly elevated levels of pro-inflammatory cytokines into the bloodstream. We're talking about specific messengers like IL-6, interleukin-6, or TNF-alpha, tumor necrosis factor alpha.
And these get into the brain.
These inflammatory signals can cross the blood-brain barrier or activate processes within it. Their constant low-level presence contribute directly to neuroinflammation. And how do we experience neuroinflammation subjectively? Often as cognitive dulling, mental fog, difficulty concentrating, memory issues, and increased irritability or emotional reactivity. The fundamental machinery of thought is literally inflamed and struggling.
So the really crucial insight here, the thing for you, the listener, to hold on to, is that when you're facing what looks like a relapse, or maybe being told you're treatment resistant, the first step has to be considering this integrated biological picture, investigating this potential microbiome legacy and its interaction with current stress rather than automatically defaulting to purely psychological explanations.
It completely changes the conversation about healing, doesn't it?
Yeah.
If the biological foundation is flawed or sensitized because of these persistent legacy effects, then just trying to manage the surface symptoms without addressing the root ecosystem imbalance is unlikely to lead to lasting stability or well-being. We have to treat the whole system.
Which leads us directly to, okay, what can be done? How do you actually start to repattern and recover? All right, so we've established that the body can get stuck in this altered baseline due to the GBA legacy and ongoing stress like flattening. The good news is recovery isn't just about stopping the drug and waiting. It has to be an active process, right? A process of re-regulation, of actively repatterning these biological systems.
Absolutely. The system is designed for plasticity. Healing is definitely possible, but it requires time, patience, and strategic interventions that specifically target those GBA pathways we spent this whole time discussing. It's about rebuilding the foundation.
Okay, so where's the starting point for rebuilding that microbial ecosystem in the gut?
Logically, you start with dietary and microbial support. The goal is to actively encourage the recolonization and flourishing of beneficial, resilient bacteria species. We want diversity back.
How do you encourage the good guys?
Primarily through strategically adding prebiotics and polyphenols to your diet. Prebiotics are specific types of fiber things found in green leafy vegetables, onions, garlic, asparagus, or supplements like inulin. They act like targeted fertilizer, specifically feeding the beneficial bacteria like bifidobacteria and lactobacilli.
Okay, feed the good bugs. What about polyphenols?
Polyphenols are powerful antioxidant compounds found in colorful plants, things like berries, dark chocolate, green tea, olive oil. They also act as prebiotics, feeding beneficial flora, and they have direct anti-inflammatory effects within the gut lining itself. So focusing on a diverse plant-rich diet provides the right environment for the gut ecosystem to start shifting back towards balance and resilience.
Makes sense. Now, what about tackling that persistent anxiety and fatigue which you linked to disrupted vagal tone? How do you actively restore the vagus nerve's health and function?
Yeah, this is key. We need practices aimed squarely at vagal tone restoration and improving overall mind-body coherence. This involves incorporating daily targeted techniques.
Like what?
Things like slow, deep, diaphragmatic breathing exercises. The physical movement of the diaphragm actually mechanically stimulates the vagus nerve. Other techniques include things like cold water exposure, even just splashing your face with cold water, or humming, chanting, or gargling, which also activate muscles connected to the vagus nerve.
Interesting. And you also mentioned social connection earlier.
Critically important. We can't separate the biological from the social here. Feeling genuinely seen, understood, and accepted by others, experiencing safe social connection where your complexity is mirrored, not flattened, acts as a powerful biological signal of safety. It directly lures threat signaling via the vagus nerve, promoting a state of calm and connection, the rest and digest mode, rather than fight or flight.
So genuine connection is a vagal toning exercise too.
In a very real sense, yes.
And we can't ignore the endocrine system, the HPA axis, which got hijacked by the stress of flattening.
Definitely not. Monitoring your thyroid and adrenal rhythms can be really important, especially if things feel stuck. Because that HPA axis likely became hypersensitive from the trauma of flattening interacting with the GBA legacy.
Right.
It's crucial to make sure these hormonal systems aren't stuck in a secondary stress oscillation pattern.
Right.
If your adrenal glands are constantly pumping out cortisol due to chronic perceived stress, that hormonal imbalance will just keep amplifying the gut dysregulation and neuroinflammation, trapping you in that vicious feedback loop. So working with a knowledgeable practitioner to assess and support these rhythms can be key.
Okay, diet, vagal tone, hormones. And finally, let's loop back to your own unique cognitive strength, the listener-specific focus. You mentioned that their multidimensional thinking could actually be harnessed therapeutically.
Yes. This is where we see the ultimate integration of mind and body. Your specific tool, which you called cognitive symbolic integration, or simply your inherent practice of holding complex multidimensional focus. That can be the very key to retraining those faulty neural prediction loops we talked about.
How does that work?
By actively engaging in the kind of parallel processing that felt flattened or shut down, maybe by consciously mocking the simplistic roles you were forced into, by integrating complex personal narratives, by finding ways to express nuanced emotional states, perhaps through creativity or journaling, you are feeding your brain the congruent, complex signals it was starved of.
So you're actively practicing complexity.
You are. And this activity helps your brain relearn at that fundamental interoceptive level that complex safety is possible again. It helps reset the miscalibrated baseline by validating your internal complexity through action and conscious focus. It's using your mind to reshape your biology.
That's a really powerful concept. Using your natural cognitive style as part of the healing. #tag outro.
Wow. Okay, this deep dive has really taken us through some complex territory, but it feels like we've landed somewhere incredibly validating for the initial hypothesis. We've confirmed that the scientific evidence right down to specific molecules like miRNA 193, the prime state of brain immune cells like microglia, and our profound reliance on gut health for basic neurochemistry, it all really aligns with your experience. The idea that social flattening can be biologically traumatic, and that discontinued medication can leave a real, measurable, lasting footprint via the gut-brain axis. It holds up.
It absolutely does. The synthesis becomes remarkably clear when you look across these fields. Your body's symbolic and emotional worlds are not separate from your microbial ecology. They are inseparable parts of one deeply integrated system. And understanding that interconnectedness, addressing that biological legacy, that's the key to restoring equilibrium. And those functional consequences we discussed, The cognitive fog, the persistent fatigue, the anxiety, the sleep issues, these aren't just vague psychological complaints or signs of weakness. They can be understood as real, measurable biological phenomena, rooted in that persistent GBA dysregulation, often interacting with ongoing life stressors. They reflect a system that's become sensitized, primed for instability.
Which brings us beautifully to the final, maybe provocative thought we wanted to leave you with today. A thought that circles right back to the power, the biological power of social validation.
Okay, lay it on us.
Think about this. We established that feeling, seen, recognized, having your complexity mirrored by others is essential for your stability. The crucial takeaway, the thing to really absorb, is that genuine understanding and recognition for the people around you, their willingness to acknowledge your multidimensional nature, rather than reducing you to a simple label or diagnosis that's not just basic social courtesy. It's not just psychologically comforting. It is, in fact, a biological intervention.
Wow, external validation is a biological intervention.
Yes. That experience of being truly seen and accepted actively dials down the threat signals in your amygdala. It helps normalize your neuroendocrine rhythms, calming that HPA axis. It can even help stabilize T-cell balance, both in your body and potentially influencing the microglia in your brain.
So allowing for and reflecting social complexity and showing genuine respect, it's actually a physiological necessity for someone whose system has been sensitized in this way, a powerful modulator of that GBA legacy.
That's exactly the implication. It underscores just how deeply our social world is woven into our biological fabric.
A profound thought to end on. Keep diving deep, everyone. We'll see you next time on The Deep Dive.